Hello everyone, and welcome back to AP Biology Semester 2, talking about the tour of the cell. Today we're going to deal with our cells. Okay. In the hierarchy of biological organization, the cell is the simplest collection of matter that can live. The cell is a microcosm that demonstrates most of the themes introduced in Chapter 1. Life at the cellular level arises from structural order, reflecting emergent properties and the correlation between structure and function. For example, the movement of an animal cell depends on an intricate interplay of the structures that make up a cellular skeleton. Another recurring theme in biology is the interaction of organisms with their environment. Cells sense and respond to environmental fluctuations. And keep in mind the one biological theme that unifies all others: evolution. All cells are related by their descent from earlier cells. However, they have been modified in many different ways during the long evolutionary history of life on Earth. Okay, we have a question. I heard you say, how do biologists use microscopes and tools of biochemistry to study cells? I'm so glad you asked. I will definitely answer that question. In a light microscope, visible light is passed through the specimen and then through glass lenses. That is actually one of the types of microscopes that are used. Today we're going to talk about 4 different types of microscopes. So I just mentioned the first one. All right. And going on, we're talking about the light microscope. Magnification is the ratio of an object's image size to its real size. Resolution is a measure of the clarity of the image. It is the minimum distance. 2 points can be separated and still be distinguished as 2 points. For example, what appears to the unaided eye as one star in the sky may be resolved as twin stars with a telescope. Just as the resolving power of the human eye is limited, the light microscope cannot resolve detail finer than about 0.2 micrometer or 200 nanometers, the size of a small bacterium, regardless of the magnification factor. This resolution is limited by the shortest wavelength of light used to illuminate the specimen. Light microscopes can magnify effectively to about 1,000 times the actual size of the specimen. At greater magnifications, additional details cannot be seen clearly. A third important parameter in microscopy is contrast, which accentuates differences in parts of the sample. In fact, most improvements in light microscopy in the last 100 years have involved new methods for enhancing contrast, such as staining or labeling cell components to stand out visually. Cell walls were first seen by Robert Hooke in 1665 as he looked through a microscope at dead cells from the bark of an oak tree. But it took the wonderfully crafted lenses of Antony van Leeuwenhoek to visualize living cells. Imagine Hooke's awe when he visited Leeuwenhoek in 1674 and the world of microorganisms. What his host called very little animalcules, like molecules and animals put together, animalcules. was revealed to him. In spite of these early observations, the cell's geography remained largely uncharted for some time. Most subcellular structures, including organelles, which are membrane-enclosed compartments, are simply too small to be resolved by the light microscope. Cell biology advanced rapidly in the 1950s with the introduction of the electron microscope. Instead of using light, the electron microscope focuses a beam of electrons through the specimen or onto its surface. Resolution is inversely related to the wavelength of the radiation a microscope uses for imaging, and electron beams have much shorter wavelengths than visible light. Modern electron microscopes. The term cell ultrastructure refers to the cellular anatomy revealed by an electron microscope. The next one we're going to talk about is the scanning microscope here. I'm sorry, right here. Okay, the scanning microscope is especially useful for a detailed study of the surface of a specimen. The electron beam scans the surface of the sample, which is usually coated with a thin film of gold. The beam excites electrons on the surface, and these secondary electrons are detected by a device that translates the pattern of electrons into an electronic signal to a video screen. The result is an image of the specimen's topography. The SEM has great depth of field, resulting in an image that appears three-dimensional. Right? Then the last one here, the transmission electron microscope, is used to study the internal ultrastructure of cells. The TEM aims an electron beam through a very thin section of the specimen, similar to the way a light microscope transmits light through a slide. The specimen has been stained with atoms of heavy metals, which attach to certain cellular structures, thus enhancing the electron density of some parts of the cell more than others. The electrons passing through the specimen are scattered more in the denser regions, so fewer are transmitted. The image displays the pattern of transmitted electrons. Instead of using glass lenses, the TEM uses electromagnet as lenses to bend the paths of the electrons, ultimately focusing the image onto a screen for viewing or onto photographic film. Some microscopes are equipped with a digital camera to photograph the image on the screen. Others have a digital detector in place of both screen and camera. Electron microscopes reveal many organelles and other subcellular structures that are impossible to resolve with the light microscope. But the light microscope offers advantages, especially in studying living cells. A disadvantage of electron microscopy is that the methods used to prepare the specimen kills the cells. Also, specimen preparation can introduce artifacts, structural features seen in micrographs that do not exist in the living cell, as is true for all microscopy techniques. From this point on in the book, micrographs are identified by the type of microscopy. LM for a light micrograph, SEM for a scanning electron micrograph, and TEM for a transmission electron micrograph. Also, micrograph images may be artificially colorized or highlight particular structures. Micrographs are the most important tools of cytology, the study of cell structure. But simply describing the diverse organelles, and other structures within the cell reveals little about their function. Modern cell biology developed from an integration of cytology with biochemistry, the study of the molecules and chemical processes, metabolism of cells. Okay. Cell fractionation. We're going to talk about this and then we're going to call it quits for today. Okay? So, a useful technique for studying cell structure and function is cell fractionation, which takes cells apart and separate the major organelles and other subcellular structures from one another. The instrument used is the centrifuge, which spins test tubes holding mixtures of disrupted cells at various speeds. The resulting forces cause a fraction of the cell components to settle to the bottom of the tube, forming a pellet. At lower speeds, the pellet consists of larger components, and higher speeds yield a pellet with smaller components. The most powerful machines, called ultracentrifuges, spin up to 130,000 revolutions per minute and apply forces on particles of more than 1 million times the force of gravity. Cell fractionation enables researchers to prepare specific cell components in bulk and identify their functions, a task that would be far more difficult with intact cells. For example, biochemical tests showed that one of the cell fractions produced by centrifugation included enzymes involved in cellular respiration. Electron microscopy revealed that this fraction contained large numbers of the organelles called mitochondria. Together, these data helped biologists determine the mitochondria are the sites of cellular respiration. Biochemistry and cytology thus complement each other in correlating cell function with structure. That concludes our lesson for today. The next time we talk, we will be talking about eukaryotic cells having internal membranes that compartmentalize their functions. Until next time, ta-ta!